far-removed continents in black Africans and gay New Yorkers by 1978. These were precisely the populations that received the first hepatitis B vaccines produced in Lytton's chimpanzees just 4 years earlier. By the way, the man sitting to Dr. Robert Gallo's right is Dr. Jonathan Mann. At the time, Dr. Mann was the AIDS czar for the World Health Organization. Not long after Dr. Mann witnessed my exchange with Gallo, he quit his most esteemed position, saying, quote, "Far more than a medical problem, AIDS is a socio-political imposition," end quote. If I am right, and these documents are legitimate, then generally trusted vaccines, the sacred cow of public health, given mostly to our nation's youth, said to have contributed most to conquering infectious diseases, are alternatively the world's worst curse and nightmare. The third and final document in this stunning sequence comes from the same National Cancer Institute and National Public Health Service report. Here is the historic account of American biologists and virologists transferring the most sophisticated bioweapons of mass destruction to Soviet scientists during the Cold War, even though, as Dr. Kano accurately states, the Russians were developing their biological weapons programs at rocket speed. These documents provide conclusive evidence that the NCI's molecular biologists, including Dr. Robert Gallo, traded America's most advanced secrets in bioweapons production with precise methods and materials for cancer induction, genocidal weapon preparation, and bacteriological and virological bioterrorism with their Russian counterparts and others during the Cold War.
We had developed a fairly effective biological weapons program, but the Soviet Union, exploiting the revolution in genetic engineering and molecular biology that happened in the 1970s and 1980s, went 10, 20, 30 years beyond what we ever achieved in the sophistication and lethality and variety of biological weapons.
In laboratories like this one, Vector in Siberia, Soviet scientists were able to use modern biotechnology to combine traits from several organisms into one, even to create entirely new diseases unknown to nature.
There's a guy named Sergei Popov who did this research for a long time for the Soviets, and you know, what he did was scary. He scares himself, and yet at the same time, you see, when you talk to him, you see him still fascinated. You know, he can't help himself. You know, it's like Oppenheimer and the atom bomb. You know, it's so technically sweet. Well, you talk to Sergei, and he is still infused with this sense of possibility. You know, gosh, if I just took one from column A and one from column B and one from column C and put them together in a new way, eureka, you know, what, you know, crazy things could we concoct? And some of them are crazy. Some of them are terrifyingly crazy.
I was in charge of a relatively large team, the biggest team probably in the history of the Soviet Union dealing with synthetic DNA.
Sergei Popov is now working in Virginia under a U.S. Defense Department contract to do biomedical research.
Right now we protect people from biological agents, but in the former Soviet Union we've developed for more than 15, 17 years, we've tried to develop more dangerous biological agents. The process is quite similar. It could protect people and it could be used to create more dangerous biological agents.
Popov is now studying how to help the human immune system fight disease. But as a bio-warrior, he turned the immune system into a killer. He did it with genetic engineering.
So these are bacterial cells. They are ready to take up new genetic information which is encoded in plasmid DNA.
A plasmid is a small ring of DNA found in many bacteria. This DNA provides instructions for certain traits, including production of toxins or resistance to antibiotics. Each trait is encoded in a segment of DNA called a gene. This clear liquid contains gene fragments.
So we mix this small amount of DNA with cells.
Using techniques developed since the 70s, scientists are able to take genes from one organism and transfer them to another. If done properly, the inserted gene will take up residence within the DNA of its new home. In this demonstration for NOVA, Dr. Popov transferred an animal gene, one that makes a sea anemone red, to a colony of bacteria, which turns red in the process.
Essentially, we've used the same technique in the former Soviet Union to create new biological weapons, new biological agents with essentially new properties and higher virulence.
In the Soviet Union, Popov isolated a gene that produces the protein myelin, which helps conduct nerve signals. He extracted the myelin gene from a small mammal and inserted it into the bacteria that causes Legionnaire's disease.
One of his projects at Oberlinsk was to take Legionella, this sort of mild flu bug in humans, and make it worse, right?
It's better to say it's mild pneumonia.
Mild pneumonia, yes,
yes,
like flu-like pneumonia, yeah.
It's pneumonia-like symptoms,
yeah.
And the agent which incorporated new genes was capable to induce absolutely new symptoms, which resembled in animals multiple sclerosis.
Exposed to Popov's genetically engineered bacteria, rabbits became ill with Legionnaire's disease. Then something strange happened. Their immune systems attacked the myelin around their own nerves, as if it were an invading pathogen.
The majority of animals died of unusual symptoms because pneumonia went away. At the time symptoms developed, there was no bacterial agent inside the animals.
So if you were, a physician trying to figure out what happened,
that would be impossible to find any kind of disease.
No trace.
So the animals were completely healthy, but several weeks later, it's better to say 2 weeks later, first symptoms appeared, and those symptoms were severe paralysis and final death.
The terrifying thing for me as a science reporter is that he turned this theory of bad bugs, of designer bad bugs, and made it real.
Knowing, for example, that American military labs created AIDS, the Russian military and Soviet media had a field day and the opportunity to produce this comic. This was widely circulated in Russia during the 1980s. Here, American biological weapons contractors working for the National Cancer Institute are depicted exchanging a test tube full of AIDS viruses with a U.S. military officer who pays cash for the bioweapon.
Our worst fears were confirmed in 1979 when an accidental release of anthrax occurred at a biological research facility in the town of Sverdlovsk. Much of our recent knowledge about their joint military and civilian program comes from a Soviet defector, Dr. Ken Alibek, formerly known as Dr. Kenachin Alibekov. He was the deputy director of BioPreparat, a cover organization for the civilian bioweapon and production facilities.
Meet the CIA's leading bioterror propagandist, who, along with our Grand Dr. William Patrick III, officiated the development of the world's deadliest anthrax.
Dr. Ken Alibek is a Soviet Union weapons defector who provided much information about the biological weapons programs in his country. He has indicated that his country and the United States held similar studies trying to engineer tularemia strains that would be resistant to antibiotics and vaccines.
Since his defection to America, Dr. Alibekov, alias Ken Alibek, has authored numerous articles and even a book to spread, like this government film, propaganda and bioterror. In essence, he is a pawn in a global war game featuring fear and educational indoctrination along with terrorization of the population.
Although we